So I posted this the other day. So this shows where you stand in the class. And you just take the 2 scores you got from me, add them up, and then add the highest of the score you got from Dr. McFadden, and you'll see exactly where you stand in the class. That's all there is to it. Very straightforward. And this is linked on the schedule page, so it's there for you to see. Oh, what's that? Sure, so you can see where you, but this is the basic range of where you are. You can see where you are, yeah, sure. So how's everybody doing? How's your prep coming? How's your note card coming? Haven't started, yeah. I recommend doing the note card closer to the end, although organizing it, you can start with a piece of paper or something and get that going at this point. Good? Everybody's ready for it? Nobody's ready for it? Thoughts, perspectives? Yeah. Final will be set up exactly as the midterm, and it will be a little longer. It probably won't be double because we don't have that much time to grade and get these things together. So I haven't actually finished writing it. I'm in the process of doing that right now. But I'm expecting to make it about 150 points. The distribution of grades will be 20% will be Dr. McFadden's first exam, 20% will be my second exam, 20% will be my third exam, and 40% will be the final. So the final is worth the equivalent of about 2 of the midterm exams.
Do you expect the same level of depth or detail on the final as we have in the midterm?
Should you expect the same level of depth or detail on the final as you do in the midterm? I would say the answer to that is yes. Yeah. Which means you're going to write really...
on the last couple of weeks, or will it be pretty general?
So the question is, will it be weighted more heavily on the last couple of weeks, or will it be more general? So I try to make things proportional to the coverage in the class, and I would say that means roughly that there'd be about a third, and a third if we compare exam 2, 3, and then the new material since then. I'm calling exam 2 my first exam. Yeah. And as I said in class, I won't ask you on anything before when I started teaching the class. So the final will not include Dr. McFadden's stuff, because I have no idea what he covered. Yes?
Do you curve the final or the class?
So the final, what I will do when I go to make grades is I will, in fact, lay out the grades and see how they distribute themselves. I do exactly this for that. One of the things I will tell you I don't do is I will not show you the final curve. And the reason for doing that is not to be mean, but I find that all that ever does is upsets people. Oh my God, I got within 3 points of an A minus, da 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 da. It's better not to know. So I won't show you. I seriously won't show you. I will check your grade if you're concerned about anything like that, but I won't show you the distribution of the final grades. And like I said, it's not to be mean, but I have never ever seen anybody look at that and be happy with it. It's like, why should I show anybody the distribution? And everybody wants to know, how close was I? It's like, why do you want to know that? Yeah?
Are you going to post the final grades on your website, or do we need to come pick those up?
So I'll post your final grades in your student account. Yep. And yes, you can come pick up your exams after they've been graded if you wish. And they'll be in the main office as the other ones were. Finals in the usual place, quarterly 11:09 at noon on Thursday. So we done with the review session now? So anyway, that's the sort of logistics of the final and everything related to that. So I try to make things, general. I try to make things. What I won't do is I won't go into any more depth than I went into in the material in the class. And while I don't recommend people study old exams, because I think that's just a dumb way to study, you should use the old exams, that is my exams, as outlines for your studying. So the topics that are covered there will be topics that you will see on your final. I'm not going to go pick out esoteric things that I didn't give you exams on or didn't test you on before. Okay? But the topics that are there, you will see on the final. So that's why I think the old exams that you have are useful. But I really caution against using the old exams as a study guide. I've seen too many problems with students doing that. They want, how many old exams can I lay my hands on? It's like, well, you know, what happens when one of the reasons I caution against that is I see students will go and they'll get a bunch of old exams and they'll memorize answers. That's what they will do. And they don't memorize questions. And a question changes slightly, and then they've got the answer. They put the answer down, and then they look at the final, or they look at the actual question that was there, and realize that they really got it wrong. And that's because they weren't paying attention to what the question was about. So that's why I really recommend staying away from old exams. I think they're not a good idea. As I always say, the material doesn't change. Exams do change. So remember that. Okay? All right. So what do you want to know? I've already given you all the answers. The only question is, what are the questions? And I haven't even made those up yet, so I don't even know myself. Yes?
Can you talk about the difference between operon and a promoter?
Okay. Difference between an operon, a promoter, and an operator. Very good question. Okay. So there's 3 terms that I think you should know what they are. Okay. So what is an operon? An operon is something that we find in bacterial cells. We don't find it in eukaryotic cells. And the definition that I gave you for an operon was that it was a collection of genes under the control of a single promoter. So in bacterial systems, we'll have one promoter followed by a series of genes. So that when the bacterium goes to transcribe, they will transcribe that entire thing as one big messenger RNA. So you saw the tryptophan operon. You saw the lac operon. Okay? The lac operon had 3 genes. The tryptophan operon had 10 genes. And that's how they're made in the bacterial cells. Okay? A promoter. What is a promoter? A promoter is a region of DNA whose sequence is recognized by transcription factors. Okay? That in bacterial systems, that could be something as simple as an RNA polymerase. In a eukaryotic cell, it will not be a polymerase. It will be a bunch of accessory proteins. I showed you TF2D and proteins like that that bind to promoter areas and help the RNA polymerase to bind. Okay? The promoter is a sequence of DNA that's recognized by transcription factors. That's what it is. Okay? And the last you asked about was the operator. So the operator is a term that we used specifically for the lac operon. And I used the term operator sort of synonymously with promoter in that operon. It's technically not that, but for our purposes, I use the term operator and promoter in the lac operon as essentially the same thing. I hear music. You guys hear music? Mm-hmm. Whew, I was worried. Does that help? Yes?
For beta oxidation,
Mm-hmm.
polyunsaturated fatty acids.
Yep.
Go through.
Beta oxidation of polyunsaturated fatty acids. Yeah, let's go there. So, and fatty acid oxidation. There you go right there, right? Lovely figure. And that thing isn't even very isn't even very focused, is it? All right. Well, let's think about this. I really showed you two scenarios. I showed you a scenario where you had a fatty acid that was being oxidized that had the double bond between carbons 3 and 4 in the cis position. Okay? And I showed you that there was an enzyme that can convert that cis 3,4 fatty acid to a trans 2,3. Let me see if I can enlarge that a little bit. Does that help? Yeah, that helps some. Okay. So here's enoyl-CoA isomerase, which is the enzyme that can do that. And what it's doing is it's converting a 3,4 to a 2,3. If that's the only double bond that's in that fatty acid, then you've got it all set, because this 2,3 now can be oxidized in regular beta oxidation, because water would add across that. You would then oxidize the hydroxyl group, and then you would do beta thiolytic cleavage to break off the acetyl-CoA. So at this point, this guy is ready to go through oxidation. And so that's what's happening. We've got a polyunsaturated, so we've got to consider the other one here. This one's going to go through standard beta oxidation. So we're going to have addition of water, hydroxyl that will go on here, oxidation of that to a ketone, then we're going to break off those two carbons. That's going to leave us, that's why it says one cycle of beta oxidation. That's going to leave us with this structure right here. Okay? So this structure actually starts the next round of beta oxidation. And the first step of the oxidation is the removal of the protons and the electrons from positions 2 and 3, giving you a trans double bond. And that leaves us with this structure right here. Well, it turns out this structure right here can't continue through beta oxidation as it is. And so, consequently, this system of two double bonds has to be converted into one double bond. And that's the job of this enzyme right here, 2,4-dienoyl-CoA reductase. I think I said you didn't need to know the name of that enzyme. But it's giving us a substrate that is identical to the substrate that you saw up here at this point. Okay? Identical. That is cis 3,4. And if you look at this guy right here, it's a cis 3,4. Well, that's a substrate for enoyl-CoA isomerase again, which means it's going to convert a cis 3,4 into a trans 2,3, and then that's going to go through beta oxidation. So that's really all there is to that. Okay? So if I have a poly, I've got to think about how that second one is going to get oxidized, and that's what's happening here. The simple situation is if I have a mono, as you saw before. Does it make sense? Does that clarify anything?
The only thing that's confusing is if you scroll up a little bit,
Okay.
it shows that you get rid of the first double bond.
Getting rid of this,
Right.
that's the oxidation, yeah.
But then there's another double bond that is beneath it. If you go down
Here?
right there,
Yeah,
yeah.
so you're starting a new cycle of beta oxidation. So when you go through beta oxidation, the very first step is that you've got the acyl-CoA dehydrogenase that takes away the protons and the electrons from positions 2 and 3. That's just regular beta oxidation. Okay? So that's what's happened here. So the next round of beta oxidation is happening. Remember, these are breaking in rounds. So each round we take off 2 carbons. So we're starting that next round right here. It's just that we can't get past the first step. The enzyme that handles the next step won't touch this thing right here. So it's got to be rearranged so that the enzyme will touch it. Make sense? And that's all that's going on with the rest of it. So all that we're doing and going from right here down to right here is just getting a substrate that the beta oxidation enzymes can handle. That's all we're doing. I think I said you didn't need to know the name of 2,4-dienoyl-CoA reductase, yeah. Yeah, Paul.
Would you have to know the acetyl-CoA?
Oh, acetyl-CoA, yes, definitely. You're talking about the acyl-CoA dehydrogenase? Yeah, acyl-CoA dehydrogenase. Yeah, I gave you a list of enzymes that you needed to know there. Yeah. Yeah. Yeah, so there's really You don't need to know anything more than the intermediates I talked about in class. Okay, so if you look at the urea cycle, right here, okay, what did I talk about? I briefly, large, ooh, too big. All right. I briefly talked about arginine, because arginine was needed to make urea. Obviously, urea is an intermediate that's there. I talked about how fumarate was produced, and I talked about citrulline. Those were the intermediates that I talked about in the urea cycle. I said you didn't need to regenerate the cycle. You're not going to do that, no. I'm sorry?
I didn't know you had mentioned citrulline. I thought it was.
I talked about this being in energy drinks.
Right, and then I thought you mentioned the next one.
Oh, it's spartate. Well, spartate is actually donating things that are there. Yeah, you're right. So that's correct. Yeah,
So,
Olivia.
noted in the urea cycle that because it occurs in the cytoplasm and the mitochondria, that there's a shuttle system. Do we need to know any details about that?
I haven't talked about the details of the shuttle, so it's like always. I haven't talked about that in the class. Yeah. But anytime you go across a membrane, it's going to take some kind of a shuttle system to make that happen.
While we're on this topic, can you go over the regulation of the purine and pyrimidine?
Sure, sure. So go over the regulation of the purine-pyrimidine biosynthesis. So I talked about, at the level of the purines, and by the way, there are other regulatory systems besides the ones I talked about, so these are not the only ones, but these are the ones that you're responsible for. So I talked about how that the synthesis of the purines, the AMP and the GMP, proceeds along the same lines and produces the same intermediate. So they start in the same place and get to right here. And when it gets to IMP, which is inosinate is what it's called, when it gets to IMP, there's a branch, one branch going over to make AMP and the other branch going over to make GMP. I pointed out that there were really two ways here that this system was balanced. One is that this enzyme, whose name you don't need to know, is feedback inhibited by AMP. So if AMP concentrations get too high, this enzyme will be turned off. Similarly, the IMP dehydrogenase, which is the enzyme over here, which again you don't need to know the name, was feedback inhibited by GMP. So each of these two products inhibits its own synthesis, and that's important. The other thing that helps to balance this is that the energy source for making AMP comes from GTP, and the energy source needed to make GMP comes from ATP. So what that means is if you have an excess of ATP or you have an excess of GTP, it will drive the synthesis of the other one. And so again, as I talked about in class, balance of these nucleotides is so important. We get too much of one or too little of the other, and the cell is much more prone to have mutation. So that was what I talked about with respect to the regulation between the individual purines. Okay? With respect to the pyrimidines, I talked about, I showed you this pathway, and I pointed out that this enzyme here called ATCase is a very critical enzyme because this enzyme is inhibited by the end product of the pathway. And this pathway doesn't stop here. It actually goes on and ultimately makes CTP. So CTP is the end product of the pyrimidine biosynthesis pathway. And CTP inhibits the enzyme ATCase, which helps ensure that cells don't make too much in the way of pyrimidine nucleotides. All right? The flip side of it is that the enzyme itself is actually also activated, and it's activated by ATP. and ATP is a purine. So this enzyme plays a very critical role in balancing the relative amounts of purines and pyrimidines, and it does that just by the scheme that I just described to you. Too much pyrimidines, turn it off. Too much purines, turn it on. And though I didn't talk about it in class, I'll mention it to you here, and it sort of stands to reason. In my BB450 class, I talk about how the decision to go through division, for example, is a very big one. Cell division is a very big one for cells to make. It takes a lot of a cell's resources to make that happen. There's a lot of nucleotides it has to make, proteins it has to make, and so forth in order for that to happen. So the cell needs to have some sort of a measure about whether it's ready to do that or not. But one of the ways in which it has that measure is the amount of ATP that it has. ATP, of course, is the energy source of the cell. If the cell has abundant ATP, then it is much more likely to be prepared and ready to do all the things necessary for division. And ATP is also turning on this enzyme, and it's making things that are needed for cellular division. So it's kind of a cool balance that's there that the cell has that ensures that it is ready for division and that it's going to make the right amounts of the individual nucleotides. Okay? Yes, sir. Okay, so factor-independent termination and how the hairpin forms. So we're talking about a termination of the process of transcription. And when I talk about transcription, protein synthesis, it's been a while, hasn't it? RNA synthesis, here we go. Come on. There we go. Chain termination. So factor-independent termination happens in a manner like what's shown right here. So your question is, how does this thing form? All right. Well, it turns out this thing forms really readily. All right. If I take two sequences that are complementary to each other, and I just put them in solution, they will find each other. Even if they're completely separated, they will find each other. It's one of the things that I describe as the sort of magic of molecular biology. When I talked about the polymerase chain reaction, for example, I talked about how we use primers to start DNA replication, right? Well, those primers have to find the complementary sequence on the DNA that they're going to amplify. And the fact that they do, obviously, it should convince us that finding each other is not a difficult thing for these sequences to do. Now, why do I say that? Well, I say this because when we look at this sequence right here, what we're looking at is the front end of an RNA, a messenger RNA that's being made. The DNA polymerase is about right here, and it has finished synthesizing this entire sequence. Okay? So it turns out that for factor-independent termination has two sequences like this that will form base pairs with each other. Well, in this case, the base pairs aren't even on separate pieces of DNA or RNA. They're within the same one, and they're only a few base pairs away. They have no trouble whatsoever finding each other. So once they've been synthesized, they immediately find each other. And when they find each other, they form the base pairs that you see right here. And the finding each other also provides what I like to describe as the equivalent of an automobile car jack. All right? And like a car jack will lift up the end of a car so that you can change the tire by getting the tire off of the ground, this jack lifts up the end of the RNA polymerase, which is sitting right here. The RNA polymerase is right here. So this thing physically comes into being. It lifts up the RNA polymerase, and since the RNA polymerase is not held to the DNA that it's copying very tightly at all, that lifting is sufficient for the RNA polymerase to just get kicked off of the DNA. When it does, that's why I say everything falls apart. The RNA polymerase falls off, the RNA itself falls off, and you'll notice that there's only UA base pairs that are holding the thing together in the first place. So it's very easy for this whole thing to fall apart, and it does. And so with factor-independent termination, we see quite specific places where termination occurs. That help? Good. I hear all the allergies going on. It's just awful right now what the pollen count is, isn't it? Question? Yep. Sure. A, P, and E, yeah. So the question is, in the elongation phase of translation, can I explain the sites and what's happening at those places? So let's take a look at protein synthesis, and let's look at what's happening during this elongation process. And I am going to try to zoom in on that. Didn't change it much, did it? Okay. So we have the ribosome, which has been assembled on the messenger RNA. That assembly happened during the initiation phase, and it resulted in the formation of 3 sites within the ribosome: an A site, a P site, and an E site. You might say, well, how come it wasn't there before? Well, it essentially was. The A site was part of the upper and part of the lower. The P site was part of the upper, part of the lower, and the E site was part of the upper and part of the lower. And when you put them together, you get a complete A site, a complete P site, and a complete E site. That's what's happening. The P site is the place where the transfer RNA is located that has the polypeptide attached to it. If we've just finished initiation, it has only a methionine. Okay? So what happens next in the process is that this protein factor called EFTU, which, as I said, is the most abundant protein that's found in E. coli. EFTU is this protein that covers up the bond between the transfer RNA and the amino acid. And it carries that complex of the tRNA linked to the amino acid into the A site. The A site is open when we start this process. So in the A site, there is a codon, and on the tRNA, there's an anticodon that might be complementary to it if the right tRNA has been brought in. All right? If the right tRNA has been brought in, then it'll have an anticodon that will be complementary to the codon that's in the A site. If it is complementary, EFTU actually checks to see that it forms the right base pairing that's there. If it sees the correct base pairing, it says, okay, I've done my job, and EFTU goes flying away and leaves these 2 guys, these 2 lovebirds, as it were, in the A site and the P site. Okay? Now, when it goes flying away, it leaves it within the confines of the ribosome, so there's no water that comes in there and releases that amino acid. After the proper amino, and if it doesn't have the right one, let's say I brought in the wrong tRNA, because these things don't form a proper base pairing, then EFTU will take it away, and a different one will come in. And so until the right one comes in, nothing will happen. Anyway, so what happens at this point then is that we've got, when we finally have the right one in there, we have something in the P site that has the polypeptide chain, and we have something in the A site that's the new amino acid that's linked on there. The next step in the process is to join that new amino acid to the rest of the polypeptide chain that's already there. And that's the formation of a peptide bond. And that formation of the peptide bond is catalyzed by the 23S ribosomal RNA. Okay? It's catalyzed by what we call a ribozyme, and that's a catalytic RNA, which is the 23S ribosomal RNA. It forms that peptide bond between the two, and now everything is attached to that tRNA that's in the A site. The tRNA that was in the P site has lost the polypeptide chain, and it's getting ready to exit the ribosome. So in the next step, there is the protein factor called EFG that comes in, and it physically slides the ribosome 3 more nucleotides down the messenger RNA, so that what was in the A site moves to the P site, what was in the P site moves to the E site, that's the exit site, and now the A site is left open again. So in essence, we're right back where we started up here, and we can continue that process. Make sense? Again?
No, I'll go.
Oh, I was going to say, okay, good. Watch the video. Okay. It's a very intricate process, and needless to say, I am again shortening a lot of what we know about that process in here, but that's the headlines of what happens within it. It's pretty cool. So no, the S designation refers to the size. It's a Svedberg unit. The subunits themselves have sizes of 60S and 40S. That's the size of the complex of the proteins and the ribosomal RNAs. The 40S is the small subunit. The 60S is the large subunit, and it's the 60S that actually holds the 23S ribosomal RNA. Okay? Olivia? The Calvin cycle, yeah, sure. So the Calvin cycle is what we refer to as the dark phase, or the dark cycle of photosynthesis. And the Calvin cycle is the place where the carbon dioxide from the atmosphere is actually assimilated into, ultimately into glucose. Okay? So that process happens as a result, or it starts at least, as a result of action of an enzyme we called it Rubisco, which is ribulose 1,5 bisphosphate carboxylase, which is what its full name is, but you don't need to know that. Rubisco is what we call it. And Rubisco catalyzes the reaction by putting a carbon onto ribulose 1,5 bisphosphate and creating basically an unstable intermediate that then splits into 2 3-carbon pieces, and those 2 3-carbon pieces are called 3-phosphoglycerate. So there we go. Okay, so you can see this carbon dioxide coming in. Here's the 2 3-carbon pieces that are being made, and we proceed along this way. Now, as I said in class, this step that I showed you right here only incorporates 1 carbon dioxide. So in order for me to make a glucose, this reaction has to happen 6 times, which is why you see the 6 right here. Okay, 6 carbons get fixed, and then the rest of it is just bookkeeping, going through all these various things here. As I pointed out in class, that part of it is like gluconeogenesis. There's phosphoglycerate kinase, there's glyceraldehyde 3-phosphate dehydrogenase, there's triose phosphate isomerase. These are all enzymes of gluconeogenesis, and aldolase, et cetera, are all in here. And the upshot is that we get as a final product of this process glucose. Now, this process depends upon the light cycle of photosynthesis, because it's in the light cycle of photosynthesis that we make 2 things that's necessary for this process to occur. One thing is ATP. It takes a lot of ATP to make glucose, starting with carbon dioxide. And the second thing that we need in doing this process is NADPH. Look at the NADPHs here. Here's 12 ATPs, here's 12 NADPHs that are necessary to make this cycle go through and ultimately to make glucose. So the pentose pathway is related to this. It has many similar reactions. To make a glucose. To make a glucose. So do the math. NADPH is equivalent to NADH in terms of energy. So if we were to take 12 NADHs and run it through the citric acid cycle, through the electron transport and oxidative phosphorylation, we would get 36 ATPs, right? And we're taking another 12 ATPs here. So in order to make that molecule of glucose, it's taking the equivalent of about 48 ATPs to do that. How many ATPs do we get out of it when we oxidize glucose? Well, as I say, it depends on how we count, but typically the numbers are 36 to 38 ATPs we get out. So that means, as always, that nothing's 100% efficient. It takes more to make it than it takes than we derive from getting it out, and that's partly because heat is lost in that process. Yes, sir.
Can you go over Pribnobox and Tatabox?
Pribnobox and Tatabox. Okay. So I sort of use the terms Pribnobox and Tatabox interchangeably, though technically the Pribnobox is used to describe the promoter region for a prokaryotic system. So let me pull this up here. Okay. So if we look at the promoters that we see, the regions of DNA ahead of the start site for transcription in bacteria, and this is a system in bacteria that's here, we typically see this AT-rich region of about 6 nucleotides that's usually within about 10 nucleotides of the start. The start is shown in every case here in yellow. In some places the start is not specific, but you can see where the start sites are there. This region that is AT-rich was discovered by a man named David Pribnow and became known as the Pribnobox as a consequence of that. And it's simply, as I said, just an AT-rich region that's there. In prokaryotic systems, we have very simple promoters, typically have a couple of regions that are involved in controlling things. You saw in the case of the lac operon, for example, the operator region that had some 2 or 3 regions of DNA sequence that were important for the transcription factors to bind. Here we see 2 fairly specific ones. If we compare the sequence in the Pribnobox to eukaryotic cells, we discover that there is also in eukaryotic cells, commonly, but not always, commonly an AT-rich region as well. And that AT-rich region will typically be within about 30 to 100 nucleotides at the start site of transcription. And in eukaryotic cells, it's called the Tata box. And the sequence is very similar. In the case of the Pribnobox, the consensus sequence is TAT-AAT. In eukaryotic cells, it's TAT-AAA, as I recall. So very similar sequences. And they both serve essentially the same purpose. Make sense? Yes, sir. Okay.
So we see telomerase active in cancer cells.
Mm-hmm.
Would it be possible to telomerase?
So very good question. Question is telomeres and telomerase. Telomerase active in cancer cells. Is it possible to design an anti-telomerase drug and thereby stop cancer? There are, in fact, some drugs in clinical trials right now with that very design. And some of them have shown promise for certain types of cancer. Not all types of cancer, but certain types of cancer appear to be responding to that. So that's kind of an exciting thing. because the cancer cell basically relies on having telomerase active if it's going to divide uncontrollably. And if you can stop that ability to put telomeres on the ends of the DNA, it will eventually kill itself. So, yeah, kind of cool. One of the cautions with that, I will tell you, is that there are cells besides cancer cells that have telomerase active. So I talk about fetal cells. Fetal cells, of course, have a telomerase active because that's when your telomeres largely get made. But stem cells also have that. And so if you indiscriminately give telomerase inhibitor, you might be doing some nasty things to your stem cells, which you rely on for making a lot of cells of your body. So you've got to be a little careful with at least the design of these things. Yes?
So is it the DNA that has the telomeres and then the flip or the RNA that has the poly?
So is it the DNA that has the telomeres and the RNA that has the poly A tail? So that's correct. In eukaryotic cells, the telomeres are found at the linear ends of DNA chromosomes, and the messenger RNAs have a poly A tail at the end, only the messenger RNAs. Oh, I'm sorry. Daydreaming. Synthesis of fats? Okay. The synthesis of fat is actually, you're starting with fatty acids, or you're talking about from making from acetyl-CoA? From phosphatidic acid. Okay, so that's pretty straightforward. So if we look at fat synthesis occurs. Right here. Okay. So the synthesis of fats, is shown here. So phosphatidic acid is a compound that you've seen during the synthesis of the glycerol phospholipids, and it consists of, let me blow that up again. I apologize for the quality of these figures, but this projector I think is out of focus is the problem. It consists of a glycerol backbone. So glycerol backbone is that carbon, carbon, carbon that's right there. All right? And it has attached to its backbone two fatty acids. So there's a fatty acid there, there's a fatty acid there. And at the third position, there is a phosphate. Now, as I said, this is phosphatidic acid, and this is what is used to make the phosphatidyl compounds that we find in our membranes. So the glycerol phospholipids will all have structures like this within them, and they will typically have something attached to that phosphate. So in the case of the glycerol phospholipids, we might have phosphatidylserine, which would be this entire structure attached through the phosphate to a serine, or phosphatidylethanolamine, which would have this entire thing attached to an ethanolamine. Well, in the synthesis of fat, phosphatidic acid is the starting point, and it's pretty straightforward what happens. The phosphate is removed, as you can see here. That leaves us now with the glycerol that has the carbon, 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 with one fatty acid, two fatty acids, and the third position is held only by a hydroxyl group. Then a fatty acid is added to that, and when it's added to that, we end up with carbon, 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 fatty acid, fatty acid, fatty acid, and that's a fat. Okay? Yes? Okay. Okay. So first of all, I would encourage you to look at the key. I've talked to several students who felt like they answered it really well until I showed them on the key that they really weren't right. So let me tell you what. Let me turn this off, and I will do it on the board. Let's see. Let's do. Okay. Okay. So if I've got a reaction, A goes to B, okay, and I said I've got two possible things. One is that B over A is equal to 3, and one is that A over B equals 1/3. That's the two possible scenarios that I gave you. And I said that your friend says that this reaction can go forward under either of these conditions. And my question to you was, is it possible that your friend is correct? All right? So the answer to the question is rooted in the Gibbs free energy equation, which is right here. Okay? Now, what most students did was they looked at this and they said, well, 3 to 1, this is a positive number, so therefore it can't go forwards. All right? And the answer to that is you can't look at any one term and decide what the value of delta G is. You've got to consider all of the terms that are here. So the scenario I would give you is, well, what if the delta G0 prime, which is a constant for a given reaction, what if it's very negative? And you tell me that, well, this is 3 to 1, this is positive. If this is more negative than this is positive, then the reaction can go forwards. And if this value is 1/3 and it's negative, then of course it's going to go forward if this is also very negative. So the answer is yes, it is possible. And the reason it's possible is because this is only one component of this. And most people looked only at this component and didn't consider this component. So to answer the question and get full credit, you had to recognize this and what this was. If you didn't recognize both of those, then you didn't get very much credit, because that would be like saying that delta G is equal to this, and that's not right. And in fact, I gave you this equation in the problem so that you would look at it and think of that. Make sense? Does it make sense? Well, then that was the wrong assumption. So the question is it possible? Can you think of a way in which this happens? And so a way in which this happens would be that delta G0 prime is this, right? Because if it's possible, there has to be some way for that to happen. If you start out by assuming this is 0, then you've already reduced your possibilities. I would agree with you that if I gave you the problem and I said this value is 0, is that possible? Then your answer would be correct. But I said, is this overall thing possible? I didn't give you this value, right? So the answer is, yes, it is possible if this is equal to a large negative number. It's not possible if it's equal to a large positive number. It's not possible if it's equal to 0. But it is possible if this is equal to a large negative number. Does that make sense? Okay. Clear as mud. Yes? What is complex 5? Okay. Complex 5 is that spinning turbine in the membrane of the mitochondrion that the protons go through that cause ATP to be made. It's also known as ATP synthase because it makes ATP. Are we done? All right, folks. Well, thank you very much. I will be in my office the next 2 days. I've got meetings and so forth, but I'm there. So if you have questions, come see me. Actually, I take it back. On Thursday morning, I've got a meeting that's probably going to take most of the morning, so don't wait till the last minute and come see me. I will try to be back in my office by about 11, but I can't even promise that. But I will try to be there as quickly as I can. I will be there all day tomorrow.